Thank you, Tony, and good afternoon to you all, and welcome back for this interesting session, I think, on antibody-drug conjugates. As Tony tells us, it's taking us into the next generation of antibody-based therapeutics. I will actually say from the outset that I'm not just going to talk about PolyTherics. This is really, and I was asked to cover the world of antibody-drug conjugates in general, so I'll cover a number of technologies, a number of technology offerings. I'll also make an apology. There are many companies operating in these fields, many academic groups operating in this field, and so I can't name them all, so I do apologize to anyone who is in the field and who I don't name check with your technology today. But it's not just a promotional presentation for PolyTherics. So let's go back more than 100 years to Paul Ehrlich, who conceived the concept of magic bullet, and also conceived the term of chemotherapy, which was really the application of chemistry to the development of new therapeutics. So he was bringing the world of chemistry into pharmaceutical industry. And also the magic bullet concept that he developed is now very, very, very applicable to the antibody-drug conjugates, which I'll come on to. So we're really seeing that term embodied in products that are reaching the market today. So we've seen the evolution of antibody-based therapies. So we've seen conventional mAbs. We've talked about conventional mAbs this morning. They're enhanced mAbs with the effector function enhanced mAbs, the bispecifics, both bispecifics addressing two targets on the same cell, and redirected T cells through a bispecific approach. But I'm most interested in, and this session is covering antibody-drug conjugates. So illustrated at the top here, the antibody-drug conjugates hitting a tumor cell target. And interestingly, the next generation beyond straightforward antibody-drug conjugates is actually thinking about bispecific antibody-drug conjugates. So again, we've heard about bispecifics from John F. this morning, and we're again looking at and thinking about bispecific or multivalent antibody-drug conjugates. And at PolyTherics, we've got a collaboration with Biotechnol with their tri-body constructs to develop tri-body-drug conjugates. So that's the next generation above and beyond where we stand today with antibody-drug conjugates. So I'd like to acknowledge the FDA, actually, for this slide, and I apologize for the militaristic tone, for those of you with a more peaceful tendency. But actually, it's a good analogy to look at what is an antibody-drug conjugate. You've got your antibody, and you've got that cytotoxic payload conjugated to it. So you've got the targeting mechanism, which is the antibody, and you've got the business end, the payload, which is the cytotoxic drug. So it's a good analogy, and the FDA use this as they think about some of the regulatory questions around an ADC, because there are two parts to it, the chemistry and the biologic. So the antibody-drug conjugate itself, as I say, is effective in specifically delivering the toxic payload to a tumor. You've got the benefit of the antibody and the tumor target specificity and the toxicity of the chemotherapy. And really, a lot of the benefit comes from reducing the side effects and the off-target toxicity associated with non-targeted chemotherapy. So that's the benefit. It's expanding the therapeutic window of a chemotherapy agent by having an antibody-drug conjugate. But an antibody-drug conjugate itself is multiple components. So you've got the target, and appropriate target selection is important in developing an ADC. You've got your vehicle, which may be a full antibody, a full IgG1 or other variants, or other scaffolds. So you've got a number of different biological vehicles. So antibody-drug conjugate is a broader universe than just antibodies. For a while, we talked about protein-drug conjugates, but actually there are also small peptides being used. So it's really any biological targeting entity can be used in an antibody-drug conjugate. The linker and then the payload. All of these pieces come together to build your antibody-drug conjugate. Some would argue, and I quote Clay Siegel from Seattle Genetics, the linker is the most valuable part of an ADC. I won't disagree with him, but I'll accept others will say equally valuable are the vehicle and the payload. But certainly the linker is a significant part of the value proposition of an antibody-drug conjugate. So the antibody-drug conjugates work by targeting, circulating in the plasma, binding to the receptor, and then ultimately the ADC receptor complex is internalized, and typically in the lysosome you then see the cytotoxic agent released, leading either, depending on its mechanism of action, to cell cycle arrest or apoptosis through DNA intercalation and strand breakage. But that is the primary mechanism of an ADC. It's taking your cytotoxic drug into your tumor cell of interest through the appropriate target. So the characteristics split through different domains. There's the characteristics of an antibody, characteristics of the linker, the characteristics of the drug, which you need to address to develop your ideal ADC. So of course the antibody has to address an appropriate target. That target has to have exploitable selectivity. And as I've said, that target can go all the way through from fully human antibodies down to novel scaffolds. But remember that many of these patients will be immunosuppressed to a degree, and therefore the issues around immunogenicity and fully humanized antibodies are less critical for an ADC than in other chronic indications. Clearly, for an ADC, you need to have an efficient internalization mechanism associated with its target. Although, again, I will caveat that. There's companies such as Immunomedics who have extracellular drug conjugates, where it's bringing the payload to the extracellular domain and localizing their extracellularly. So for their targets, they're not dependent on internalization. And then obviously the conjugation shouldn't adversely impact upon the PK and the binding of your antibody. The linker itself, this is where the chemistry comes in. You've got to have an efficient conjugation process. You don't want to have a process where you're throwing away a vast majority of your antibody, a vast majority of your cytotoxic reagent going into the process. It's got to be stable in circulation. We've got to get the release, though, after intracellular internalization. And from a CMC perspective, you want to limit the heterogeneity because the regulators like that. Similarly, you limit the heterogeneity, you limit the variability of the response in terms of the PK and the efficacy that you see with your ADC. The drug itself, there are many available drugs, and I'll cover some of these, but you need a very potent drug. One of the benefits of ADCs, you can go to more potent cytotoxic drugs than would be available in a non-ADC context. So some of the picomolar potent cytotoxics are not appropriate outside the ADC domain. They are good payloads in the ADC domain. The drug itself, and I'll come on to some chemistry here, and apologies, I'm not a chemist myself, and apologies, I guess most of us are protein chemists or biologists. There's going to be a bit of chemistry, but their chemistry is important because you've got to be able to attach your drug to the linker, to the antibody. So the drug's got to be amenable to those linker attachments. As I say, the drug itself has got to be stable and increasingly a good understanding of that mechanism of action, including overcoming the multiple drug resistance. You don't want to internalize your cytotoxic drug only to have it spat out again by the tumor cell. One of the benefits, though, if you do get it internalized, is you get distribution through the tumor after initial internalization. So there's a benefit of a drug which can permeate through the tumor, and known as the local bystander effect. So all of these characteristics come into play as you're building an ADC. And this is just, and I don't think this is comprehensive, and I don't expect you to read it at the back, but more than 24 drugs are in development as ADCs today. The top one, Mylotarg, is still in development despite having been withdrawn from the market. There are many in phase 1. Roche have got more than 25 ADCs in development. But in terms of the common names you'll see in this list, if you can read it, the names of Seattle Genetics and Immunogen come through very strongly because they are two of the dominant players with their payload offering in the ADC space. The targets, and again, and the indications, I don't expect you to be able to read this full list, but there's a multitude of targets being addressed with ADCs, and these cover solid tumors as well as hematological tumors. This is an approach which is applicable to both solid tumors and hematological tumors. In terms of selecting your payload, though, there are differences. This is some data that's been presented in the past by Hans-Peter Gerber of Pfizer, and so I've taken it from his presentation, and basically looking at a range of different payloads. What you can see in proliferating cells, the mechanism, whether it's an antitubulin inhibitor or a DNA binding agent, equally effective in proliferating cells. In quiescent cells, in cancer stem cells, what you're looking for there is a drug which is equally potent, and this is where the DNA binding class comes into effect. And so without stealing the thunder from a later speaker in terms of Spirogen, yeah, a lot of their offering is in that space, so it gives us an alternative away from Imugen and Seattle Genetics payloads. So Mylotarg is one of the leading ADCs. This is using a calicheamycin payload, a glycosidic antibiotic, highly lipophilic payload, which does bind to the DNA minor groove, ultimately leading to apoptosis. It was approved for AML, got accelerated approval. However, in the post-approval study, the significant toxicities were seen, partly driven by the extreme heterogeneity of this product, and the product was withdrawn back in 2010. Although I do say that Pfizer have got a number of ongoing clinical studies looking to bring that product back to the market. One of the issues is that the conjugation is to lysines of the antibody. And so you've got an extreme heterogeneity of the product, whilst on average you'll see it says there's 2 to 3 moles of calicheamycin per antibody. You've got potentially up to 20 copies, and it's a very heterogeneous product. Adcetris is on the market, developed by Seattle Genetics with Millennium, this is the Takeda oncology company, very successfully developed by Seagen. And I take my hat off to what Clay Siegel has done in terms of developing the company and developing this product. Targets CD30, which is a target overexpressed on the Reed-Sternberg cells, which characterize Hodgkin lymphoma cells. So it got granted accelerated approval, and with ADCs, it's a very real prospect of getting accelerated approval for these products. Back in 2011 for Hodgkin lymphoma and systemic anaplastic large cell lymphoma. In Europe, it's now also got conditional marketing approval, and sales are ramping up, $100 million of sales in the first 3 quarters of last year. So that's a successful product establishing the ADC concept. You can develop, get approved, and market an ADC product. The payload for Adcetris is auristatin E. Auristatin E is a derivative of a molecule derived from the Indian Ocean sea hare, discovered by George Pettit of Arizona State University, a man who's discovered many highly potent cytotoxic drugs. Yeah, he enjoys diving. If you ever have the opportunity to hear one of his presentations, I do recommend it, certainly for the holiday shots he can show. So from diving in the Indian Ocean, he had derived the dolastatin 10 peptide, which is active as a cytotoxic, and then derivatized that to what is known as monomethyl auristatin E, because at the N-terminus of this peptide, you lose one of these methyl groups, and so it's the monomethyl variant, and that enables conjugation to a linker at the N-terminus to build your ADC reagent. So this is the rest of the reagent which gets conjugated to the monoclonal antibody. So you've got the auristatin E component, you've got a cathepsin B labile dipeptide linkage, and then you've got a self-immolative linker. So after the cleavage by cathepsin B of this dipeptide, that falls off, leaving you with the active monomethyl E. So that is the reagent which is being used for Adcetris and a number of other products in development, conjugated to antibodies via a sulfhydryl linkage and a maleimide linker chemistry. One of the challenges in developing such a product when you reduce the disulfide bonds of your antibody to expose that cysteine, sulfhydryl group of the cysteine, to create the conjugation is the heterogeneity. There are 4 available disulfide bonds, and so you can get, as well as no conjugate, 2 copies of the drug, 4 copies of the drug, 6 copies of the drug, and although it's not illustrated on this slide, 8 copies of the drug. So you're getting a heterogeneous population coming from this conjugation. And whilst you can vary the reduction conditions, you do see this variability, this distribution. E0, DAR 0, drug antibody ratio of 0, means there is no drug. That's a competitive inhibitor. And what they're seeing, and typically you will see, is about 10% unconjugated drug, which is one of the challenges of this approach. The higher loaded species here have more rapid clearance, greater toxicity, and so they are less desirable species. But you do get that distribution using reduced disulfide maleimide conjugation for an ADC. That's data from Seattle Genetics. That's data using exactly the same chemistry from Astellas Genesis group. So you do that, see that variability, and you do see typically 10% or so non-conjugated antibody going forward in your product. There are a number of variants for auristatin. So I've said there's dolastatin 10, there's the auristatin E derivative where you've got that modification there. That's the monomethyl auristatin E. And then there's monomethyl auristatin F, which has a further variation here, creating a more polar molecule. You get a reduced bystander effect. This is 3 orders of magnitude less potent in vitro in a non-conjugate form, but is as active as auristatin E as an ADC without the bystander effect, as I say. But you don't require a cleavable linker to use auristatin F, whereas you do for auristatin E typically. So there's a bit of a black art when it comes to selection of your payload. But certainly auristatins are a viable class of payloads to take forward. TDM1 is the Roche-Genentech variation of Herceptin as an ADC. So you've got Herceptin, established antibody. Let's make an ADC out of that. The BLA for this product was submitted in October of last year. Lysine conjugation again. There's 88 lysines in trastuzumab. 3/4 of them are available for conjugation. So you're going to have a very heterogeneous product coming out of this, not helped by the fact that it's a 2-step conjugation process. So you start with your mAb, you conjugate a linker group to that, and typically in TDM1 you have 5 copies of the linker per antibody, and then you add the maytansine payload, and typically you have 4 copies. Quick bit of maths, you've got 5 copies of the linker on average and 4 copies of the payload on average, meaning you've got 1 copy of the linker on average, non-conjugated. So you've got a very heterogeneous product. That being said, it has been effective in clinical studies. This is, again, phase 3 study, the Amelia study presented last year, where they're comparing TDM1 versus a load of Ty-curve combination, did see an overall improvement in median survival, 5-month extension in life expectation, and a reduction in significant side effects. So one of the important aspects of an ADC is that reduction in the side effects compared to existing standard of care. So lysine conjugation is a challenge because of the number of lysines in an antibody and also the approach to building the reagent. So if you've got a 2-step conjugation, you've got 2 degrees of heterogeneity coming into the product. But Immugen have developed a number of different linkers. So this is the SMCC linker that's used in TDM1, which then has a thioether linkage, and it's non-cleavable. And basically, the active drug is reduced by proteolytic degradation of the antibody. In this variant with the SPP linker, you do have a hindered disulfide, which after intracellular uptake is cleaved. And similarly here, you have a hindered disulfide. So there are variants around lysine conjugation, but it's all lysine conjugation, so you've got the heterogeneity of that approach. So this cartoon illustrates the issue that with lysine conjugation, you get a Christmas tree lit up, and those are flashing lights almost in terms of the variability of the product. It's a low efficiency conjugation process, non-specific in terms of the site, it's more specific for lysine, but as I say, there's so many lysines, and a high heterogeneity. Reduced disulfides where you use maleimide chemistry to conjugate to either side of the disulfide bond. It becomes semi-specific. Again, you've got the heterogeneity of that approach, and I'll come on to it, but the maleimide chemistry is unstable, so there are a number of issues in using that approach. By using maleimide chemistry, though, but trying to get site specificity, you can engineer in cysteines into preferred sites on an antibody. This is the thiomab concept developed by Genentech, but not exclusively by Genentech. Others are using this approach, where you, rather than reducing the disulfide bonds, use engineered cysteines as your site of conjugation, and then the target would be a DAR of 2. So it's site-specific. You reduce the heterogeneity of the product, but if you're using maleimide chemistry, you still have the issues of maleimide instability. What you see with the maleimide instability is that drug falls off. Now, it varies depending on the site of your cysteine. So again, this is some data from Genentech, where they've actually engineered the cysteines either into the light chain, the heavy chain, or the FC region. And what you see over the time course of incubation in plasma is essentially the DAR2, which is your starting species. You're getting significant loss of drug to create the DAR1 and DAR0 species. But it varies depending on your site of engineering your cysteine into the protein. One of the other challenges is by introducing those extra cysteines, you can get aggregation. What you can do is see it as an extra copy of the light chain conjugating, so you get heavy, heavy, light, light, light products, again reported by Genentech as one of the issues with the thiamab approach. You also see variable efficacy depending on where you put the cysteine. Again, this is data from Genentech, where if you have it on the light chain, you have a more effective product than on the FC domain. And we also see, over time, this is in cynomolgus monkeys, that you see a loss of the DAR2 species, increase in DAR1 and the DAR0 species. So you've got a product which has the long half-life of the antibody, but it's unstable in circulation. So there's a certain bioanalytical challenge there, and also the variability you'll get from an efficacy perspective with those results. And you've got to also remember that where's the drug gone? Because the drug with the maleimide linkage will, with this maleimide group, either dissociate and you get an equilibrium and potentially exchange with albumin, so you've got basically an albumin-drug conjugate, or exchange with the glutathione. So you've got an equilibrium on this side, but also with maleimide you can get the ring opening and you get this acid functionality which has issues in terms of the stability of the product. But you don't see dissociation, so that is more stable, greater therapeutic activity, but there's that issue of that that acid group leading to degradation of the product. So you've got variability in your product, both from a CMC perspective and in vivo. So World ADC Summit last year, Hanson Wade ran a survey as to what's the biggest CMC challenge for ADC products, and you can see the most significant issue is controlling the toxin-antibody ratio, and closely followed by need for improved linker technologies, and ultimately removal of unbound toxin, because also this conjugation process, you will get unbound toxin associated with your antibody. So those are the needs. So what do we offer at PolyTherics? Now I'll come to the advertising part of this presentation. So we have our ThiaBridge technology, where we can conjugate to the interchain disulfide bonds of an IgG, and I'll come on to antibody fragments as well, but basically we're conjugating to the disulfide bonds. So we can improve the homogeneity of the product and preserve the structural integrity, because what we do is those disulfide bonds, we rebridge with our reagent. So we introduce a 3-carbon linker into that disulfide bond, and those thioether bonds are more stable than the original disulfide bond, and through that reagent we then ultimately have the drug. So our reagents look somewhat like this. We have the ThiaBridge linker chemistry. We typically have a PEG linker in there, which improves the solubility of the reagents and reduces aggregation. Cleavable linker as appropriate according to the drug. And then we're working with a variety of different payloads. We don't have our own proprietary payload. We're working to build a toolkit of different payloads to enable you as a potential partner to choose the ADC of choice. It's your target. You know the chemistry. You know the biology. You choose the relevant payload. We can give you access to our reagent toolkit. The chemistry itself, if you're interested in chemistry or you're a chemist, is a simple process. Having reduced the disulfide bond, our reagent has the sulfone leaving groups, and we expose disulfide double bonds here, which is 2 Michael additions to create that species at the end, which, as I said, is 3-carbon bridge across the disulfide. We've applied it to an antibody. So this is with trastuzumab, created a DAR2 product, active in the relevant antigen-positive cell line, no non-specific cytotoxicity seen. So then we can work with antibodies, but it's not limited to antibodies. Antibody fragments are emerging as the next class of interesting targeting entities for ADCs. This, again, is part of the survey from from Hanson Wade. 35% of respondents said they see antibody fragments as a promising approach for ADCs. What we can do is conjugate to the interchain disulfide, and this cartoon illustrates that we can put one copy of the drug with our reagent through there, or multiple copies of drug through the single point of conjugation. So we can create a higher DAR species through a single point of conjugation. There you can see the reaction mixture. That's the reaction mixture in that last column. So that's a Fab, that's the reduced Fab, that's the reaction mixture of the drug conjugate. You can see how clean that is, and that's using low molar equivalents of our reagent. So it's site-specific conjugation, near 100% conversion, low reagent consumption, and this case where we've got a single drug on the reagent, a single reaction product with a DAR equals 1. And it's very easy to purify and characterize. What you see here, 99% purity of our product by HPLC. And when we challenge, so this was the Fab, reduced Fab, that's the Fab drug conjugate. Challenge in reducing environment with DTT, we see that it's stable. So we've got a very stable, homogeneous drug conjugate. Active in vitro, no non-specific cell killing with this reagent. So we've seen it's effective in vitro. And in vivo, we've taken that Fab drug conjugate into a xenograft study. Unfortunately, the xenograft study, because of challenges with the US customs, the tumors got larger than we would have liked. So it's a certain challenge we set ourselves. But what you see is the control group tumor volume escape, as you'd expect. The antibody alone, the free drug alone, you do see continued tumor growth. With our Fab drug conjugate, you see significant mean tumor volume reduction. And actually in 5 of the mice out of our group of 10, we saw complete tumor eradication with our Fab drug conjugate with every 2-day dosing. So we've demonstrated that a Fab drug conjugate is viable and active in an in vivo model. There are alternative payloads. I'll just touch upon these, and I won't steal Spirogen's thunder by talking about the pyrrolobenzodiazepines, other than to say that, as far as I'm aware, the third most potent class of compounds ever tested by the NCI. But Heidelberg Pharma offer their alpha-amanitin, derived from death cap mushrooms, which is an alternative mechanism because it's an RNA polymerase II inhibitor. And PharmaMar have got a vast variety of potent cytotoxic drugs which are derived from their marine collection. So there are a number of alternative payloads above and beyond the auristatins and the mertansins that I've talked about before. And we're looking to expand our reagent toolkit with a number of different payload offerings. There is also different expression systems. Very quickly say that cell-3 expression developed by Sutro is available. They claim to be able to produce hundreds of grams of proteins in 8 days. And what they use is click chemistry, so they engineer a non-natural amino acid into the sequence of the antibody that they express, and then using click chemistry, which has its challenges in terms of its efficiency, but click chemistry will give you that site-specific conjugation of your payload onto your antibody. And again, there are other companies offering non-natural amino acid approaches to achieve site-specific conjugation. Masana offer a variation on the reagents, which is with a polymer with multiple copies of the payload. You can see where my thinking is going in terms of some of the options for creating multiply loaded reagents. And what they've seen is that you can reduce the antibody dose and still get effective cell killing with this polymeric structure with multiple copies of the payload through a single point of conjugation. Just pause very quickly. How am I doing for time? All right, give me 2 minutes. All right, just quickly just say in terms of some of the thoughts about the regulatory considerations when you're developing an ADC. Remember, you've got an antibody and a small molecule peptide component. So both of those have to be considered and characterized as starting materials going into the development of your ADC. So they need to be appropriately characterized as a biologic and as a small molecule. And the final product needs to be characterized as both a small molecule and as a biologic. And you also need to characterize the drug-antibody ratio, the loading distribution in terms of the heterogeneity, the sites of location, and ultimately the free drug and free antibody within your product, and achieve consistent batch-to-batch reproducibility of your production process in order to have developed a product. So I go back to TDM1 and say they've got an FDA action date of February the 26th. I'd love to be a fly on the wall at the FDA deliberations around the CMC challenges with that product. But that being said, it has shown the clinical efficacy, and these products do benefit patients. So the last advert for PolyTherics, we are a broad-based technology provider enabling better biopharmaceuticals, so we're very active in the antibody-drug conjugate space, seeing a lot of inbound interest in our technology offering, where we can offer reduced heterogeneity, improved stability, and efficient chemistry, using our conjugation chemistries and working with a variety of payloads applicable to MABs, FABs, and different scaffolds, but we still do pegylation, half-life extension, and bispecifics via chemical conjugation. I thank you very much for your time this afternoon.